We have an exciting presentation for you today. Emily Martersteck, on our Senior Field Application Specialist, is going to give you a presentation on spatial mapping and in situ confirmation of gene signatures using our RNAscope HiPlex technology. This is an excellent presentation. We're really excited to share it with you. If you have any questions that pop up during the presentation, please do go ahead and ask your questions in the chat box. You can raise your hand and you can also ask a question. At the end of the presentation, we will have a live Q&A and we'll ask your questions of Emily live in person. So she's online waiting to receive those. So with that, I am going to go ahead and mute myself and I will share the presentation. Thank you.
Thanks so much, Rhonda, and everyone, welcome to today's webinar. I'll be taking the next hour or so to explain in detail how to use ACD's HiPlex assay to confirm gene signatures and spatially map cell types and subtypes of interest in the tissue morphological context. So I hope that whether you've never heard of RNAscope before or you've been using RNAscope for years, you'll get something useful out of today's talk, wherever you may be calling in from. And as Rhonda said, my name is Emily. I'm an FAS, and I'm actually based here in Boston, talking to you right now from my living room. So, outline of today's talk, I'll start with an overview of RNAscope's technology, and then we'll take a deep dive into the HiPlex assay specifically, including components of the HiPlex kit, protocol, etc. And to look at 12 RNA targets at once, the HiPlex assay requires image registration. So at the end, I'll go through a brief demo of how to use the HiPlex image registration software, which comes with the purchase of the HiPlex kit. And we'll also discuss imaging best practices for fluorescent RNAscope. And I'll be showing you some example data generated with the HiPlex assay. If you visited ACD's poster at SFN, Society for Neuroscience conference last year in Chicago, you'll recognize some of the data I'll show to illustrate how powerful and useful this assay is. But we're starting to see publications from labs using HiPlex in their own research, which is interesting. So I'll highlight a publication just out this month in Neuron from Garrett Stuber's lab over at the University of Washington. So lots to talk about today. But first, I just want to explain why this is such a useful tool. So with that, complex tissues, like central nervous system, for example, are comprised of multiple cell types and states. And things change throughout the course of development, and sometimes things go wrong. And so the ability to characterize gene expression in these tissues can enable identification of new cell types, predictive biomarkers, therapeutic strategies. I mean, there's so many interesting questions we can ask about how this all works. But we need a really sensitive, specific, flexible spatial approach with single cell resolution to deeply characterize these tissues. And traditionally, transcriptomic analysis has relied on bulk tissue analyses, such as PCR, for example, which provide an average level of gene expression in a sample. But with such grind and bind assays, you lose a lot of information. The analogy here would be there are several different colors of paint you can combine and still end up with the same color at the end of the day. But just looking at a particular hue, you don't know precisely how much of or which exact colors were mixed together. With the advent of single-cell RNA sequencing technology, now we can get molecular information at the single-cell level. So returning to the analogy, now you can see how many drops of what color contribute to any particular picture. However, these techniques still don't provide you with information about how cells are organized in the tissue context. And this is where spatial analyses come into play. With RNAscope, you have the ability to detect RNA at the single molecule level. So this is single molecule in situ, while maintaining single cell resolution and the tissue morphological context, which is a really powerful combination. So to overlay some real data on top of that analogy, we have the histogram of bulk sequencing, which is a powerful tool, but what you end up with is a sense for relative levels of gene expression in a sample respective to control genes. In the middle, there is a TSNE plot of single-cell RNA-seq data, where cells are clustered now according to their relatedness to one another. But these cells are distributed according to their whole transcriptome correlation distance, rather than how they're physically arranged in space. In the rightmost panel here, you can see RNAscope data, where now we can see the actual transcripts, the actual RNA molecules with single-cell resolution in the tissue morphological context. And I'm not the only one that's noticed this parallel between RNAscope data and pointillism, this artistic technique. So, for instance, in this featured article from Stem Cell Reports last month, researchers created this cover based on the RNAscope data that was generated from their research into the developing mouse epicardium, so epicardial gene expression. And in this article, the granularity they were able to achieve using RNAscope, in fact, refuted previous findings on epicardium-derived cell phase. So interesting, and it's beautiful to look at the same time. So a brief history lesson, because in situ hybridization as a technique has been around since it was developed in the late 1960s. And in situ hybridization is a method of detecting nucleic acid sequences in tissue by generating a probe. The probe is quite simply an oligonucleotide with a sequence complementary to your sequence of interest. And probes are labeled in some such way to allow for detection, whether it's radio-labeled probes, as in the figure on the left, or probes are labeled with something like DIG, which can be recognized by an antibody. Regardless, in situ hybridization traditionally has been really difficult. One, it takes a really long time. So, for example, the images in this 1969 paper, it took 2 to 3 months of exposure time before signal from these radio-labeled probes were visible. And the other big challenge is that RNA is an unstable molecule. Here depicted as this squiggly line, proteins and DNA are very stable. You can pull mystery tissue out of the back of a 4-degree refrigerator and successfully stain with an antibody. Unfortunately, this is not the case with RNA, because RNA in the tissue sample gets degraded over time, or fragmented potentially by fixation. And if any portion of the sequence of interest is degraded or fragmented, this probe will not be able to bind and no signal will be generated. Traditionally, these probes are quite long, 800 base pairs, maybe 500 base pairs long. So again, even if one portion is missing, the experiment will fail. An overview of the RNAscope workflow shows that the process is somewhat similar, I'd say, to traditional in situ. The assay starts with permeabilizing the sample with pretreatment reagents to unmask RNA in the sample and allow probes to hybridize to target RNA. RNAscope works whether you have formalin-fixed paraffin-embedded tissue, fixed frozen samples, fresh frozen tissue, cultured cells. The pre-treat portion of the assay here will just change depending on what your sample type is. Side note, for HiPlex, we don't recommend using HiPlex for SFPE samples because autofluorescence is an issue. We have a different RNAscope chemistry for you that I'll mention at the end of this webinar. After pre-treatment, the probes are hybridized onto the sample in a 2-hour-long 40 degrees Celsius hybridization step. All RNAscope probes behave the same way, so there's no need to optimize the hybridization time or temperature for each probe. And once the target probe is bound to target RNA, signal is detected with a series of amplification steps that build this sort of amplification tree structure on top of the oligonucleotides that make up the probe. And this generates such a high signal-to-noise ratio that RNA molecules are visible as punctate dots at the end of the day, which are quantifiable with image analysis software. And this assay can be used to visualize messenger RNA, but also long non-coding RNA, even splice variants or highly homologous sequences. So one huge improvement over traditional in situ is the RNAscope probe design. An RNAscope probe is actually composed of many oligonucleotides, here depicted as a Z, to emphasize that there are 2 regions linked here by a spacer. So the target-specific binding site here is complementary to just a 25 base pair portion of sequence of interest. So really quite short, while the top portion of the Z is the base for the amplification structure. Each Z has a partner, such that when two Zs of a pair bind exactly adjacent to each other on the target sequence, they form this pre-amplifier binding site here, onto which the amplification structure can be built. This pre-amplifier molecule is bound by amplifier molecules, sort of the branches of your amplification tree. And then there's so much labeled probe that's applied onto the branches that were attached to the pre-amplifier molecule that each RNA molecule is visible. And in the case of the HiPlex assay, this label that we apply is a fluorophore. Now, there's background subtraction built into this assay, wherein unless both Zs of a pair bind exactly adjacent to each other, you won't be able to amplify any signal because just one Z binding isn't stable enough to hold on to this amplification tree structure such that it will just get washed away. So no amplification from off-target binding occurs. The typical RNAscope probe is a pool of 20 of these ZZ pairs. So if each ZZ pair targets about 50 base pairs of sequence, and there's 20 ZZ pairs in a probe, you know that the probe is typically designed against about a 1 kb length of sequence of interest. However, you don't need all 20 of these ZZ pairs available to you to bind to your target RNA to see signal. You need a minimum of only about 6 ZZ pairs to bind. So with all that redundancy built into the probe design, it stacks the deck in your favor of being able to see signal even if the RNA in the sample is somewhat degraded or fragmented. Now, you'll notice that the probes are not directly conjugated to the fluorophore. So how do different probes end up in different fluorescent channels at the end of the day? I just want to emphasize that the top portion of each ZZ pair here that forms the pre-amplifier binding site is what determines the probe's channel or tail sequence. So all 20 double Z pairs within one RNAscope probe have the same 28 base pair sequence up here that's formed on top when the two Z's of the pair bind together. I just highlight this because the nomenclature for RNAscope probes gets a little confusing when we start talking about fluorescence because, of course, we talk about fluorescent channels like fluorescein, Cy3, and Cy5. So from here on out, you'll hear me refer to this top portion of the ZZ pair as a tail sequence for HiPlex. So dots, at the end of the day, is what you're looking at under the microscope. For quantification, we really don't care about dot size or dot intensity because the size or intensity of any particular dot just correlates with the number of ZZ pairs that we're able to land on the transcript. So it doesn't tell you anything biologically relevant or interesting to look at the dot size. You're looking for dot number if you're quantifying. And then sometimes if you have a high enough expressing gene, the dots all pile up on top of each other and start to form this more clustered signal appearance. So depending on the level of gene expression in a sample, the probe readout can look quite different. You can find more information about the probe online at acdbio.com, including the number of ZZ pairs within a particular probe, the base pair start and stop region of transcript the probe is designed against, accessed via the accession link here that takes you to the NCBI database. One note about probe nomenclature: by default, if there's nothing specified at the end of the probe's name, by default, it's a channel 1 probe. I encourage you to go online and look up information about probes before you order them, because each RNAscope probe is designed based on a customer request. You can look in the online catalog and find a probe that matches the gene target that you're interested in studying, but I encourage you to just verify that the portion of the gene the probe is designed to detect is compatible with what you're actually trying to pick up in your sample. And if we don't have a probe ready to go for what you're actually trying to look for, it takes just 2 weeks to design, manufacture, and get you a brand new RNAscope probe that's going to be very specific to the transcript you're interested in. All right, this is the moment we've all been waiting for. I'll highlight and go through the HiPlex assay specifically, how it works, why it's cool, and what it's been used for in the research context. So after the pretreatment portion of the assay, again, depending on what sample type you have, you'll do different things to your sample. But then all 12 HiPlex probes are mixed together and applied to the slide in just that 1 2-hour-long probe hide step. So by the end of that step, you have probe Z pairs bound on all of the transcripts you're interested in detecting. After that, in a series of 3 easy amplification steps, you construct 12 different and distinct types of amplification tree structure bound to ZZ pairs within all 12 of your HiPlex probes. Now, the way this works is not only do the probes have tail sequences up top here that make them distinct from one another, but with the step AMP1, you're applying a cocktail of pre-amplifier molecules, which have complementary sequences that match them to their correct probe. And likewise, in AMP2 and AMP3, you finish out all 12 flavors of amplification tree because a T1 ZZ pairs of the T1 HiPlex probe will only be bound by a T1 pre-amplifier molecule. T2 pre-amplifiers won't be able to hybridize to T1 probe. And likewise, the amplifier molecules themselves are also specific to particular pre-amplifier molecules, wherein a T1 amplifier, the branch of this tree, will be able to bind to the pre-amplifier molecule that's T1 that's bound to the T1 probe, but a T2 amplifier will not. It will go and find its appropriate pre-amplifier molecule. So this allows us to mix all 12 probes together and build all 12 amplification trees without crosstalk very efficiently all together. So at this point, you're about 7 or 8 hours into your day. And then in a 15-minute incubation step, you apply the first set of tagged fluorophores. So these are four different fluorescent dyes tagged with 4 distinct oligo sequences, such that the fluorophores will only attach to the T1, T2, T3, and T4 amplification trees, respectively. You counterstain with DAPI and cover slip, and then take an image to detect this first set of 4 probes. After that, after you've captured the first round, you gently decover slip and cleave off the fluorophore from the amplification system. The fluorophores then get washed away, but the probes all stay bound, the amp trees stay intact, and in fact, the binding site for these fluorophores is now blocked by the oligo tag that landed the fluorophores on this set of 4 trees in the first place. Because you're going to come along with your next set of tagged fluorophores, it's the same colors, but now they're tagged differently, such that they will land on trees T5, T6, T7, and T8, respectively. You cover slip and then take another image. You then repeat this process again. So to detect the final set of 4 probes, if you're doing a 12-plex, if you're doing an 8-plex, you're done already at this point. But to detect 12 targets, you do that once more. So cleavage snips away the fluorophore and washes it away. You re-fluorophore now, T9, T10, T11, and T12 probes have been detected with fluorophores. Take images from round 3 of this staining, and you're done. So as I mentioned, the probes are not directly conjugated to the fluorophore. However, the tail sequence you pick for each of your target probes does determine what fluorescent channel your probes will end up in at the end of the day. So this is not flexible. Whatever T1 probe you have will end up in green, T2 will end up in red, T3 far red, and T4 near infrared. So this is the way the default kit is set up, such that you're detecting these probes in 3 rounds with 4 colors, green, red, far red, and Alexa Fluor 750. However, if you don't have access to an imaging system that can detect Alexa 750, we have a workaround. You can instead get this alternate display module. Now, to detect 12 targets, you're doing 4 rounds with 3 colors, green, red, and far red. So it's 1 extra round of cleaving and refloring, but we do have the solution for you in case you don't have the laser lines and filter cubes for detecting Alexa 750. Okay, so just to review that workflow, after the pre-treatment portion of the protocol, you hybridize all 12 of your probes together. You build your amplification tree structures, as many for as many probes as you have. You apply tagged fluorophores that will then label these trees, take an image. And this is all done in just one single day. After you take your image, you cleave off the fluorophore, and then you repeat, you sort of rinse and repeat this as many times as you need to detect however many probes that you have. Now, at this point, you're probably wondering a couple of things. One, how on earth do you decover slip of tissue without completely destroying it? And the key is to mount with the recommended mounting media, Prolonged Gold. Prolonged Gold doesn't hard set, so you can soak in buffer these slides, and the cover slips actually float off in just about 30 minutes to an hour. Kind of amazing. You also need to make sure that you do not nail polish the cover slip onto the slide, otherwise that's going to create a problem. And then the other thing that I think always comes up for folks when they see this workflow is how efficient is this cleaving step? Because you're using the same colors over and over again. You want to make sure that from previous rounds, these fluorophores are removed completely. And then another question is often, how well does this image registration work? And I'll take you through a demo of that software in just a minute, but the cleaving step really works very efficiently. As long as you use fresh cleaving solution every time in between rounds. So this is RNAscope signal. You can see punctate dots from one particular probe that was detected in round 1 before cleavage. If you use old cleaving solution, you can see this is there's still fluorophore that remains behind. However, if you use freshly made up cleaving solution, you do have this 100% efficient removal of the fluorophore in between rounds. Here are some examples of relatively low expressing targets in fresh frozen mouse frames. This is Polar2A, T1, particle 8, MEIS1, and HPRT1. We recommend to detect your lowest expressing targets in round 1. The reason is this gives you some insurance against background creeping up throughout the course of the assay. So detect your mid-level expressors in round 2 and leave detection for your highest expressing targets in round 3 to optimize your signal-to-noise ratio. This assay is really very clean because of that background subtraction built into the probe design. But just in case autofluorescence creeps up throughout the assay, if you detect your highest expressors last, you're still totally fine. Also, autofluorescence is the worst in the green fluorescence spectrum, always, regardless of whether you're doing a protein immunostain or in situ. So we recommend to put your highest expressing target into green to further optimize your signal to noise. So here, for instance, of these 4 targets, you can see Polar2A is the highest expressing. There's the most dots and the most signal. So that one was designated as the T1 probe, so it's detected with the green fluorophore at the microscope. And the negative control probe also comes in handy. I'll talk about that more later. But this is a real RNAscope probe designed against a gene that shouldn't be expressed in any mammalian system. It's DAPB, not to be confused with DAPI, the nuclear counterstain. DAPB is a soil bacterial gene. So again, this gene is just not expressed in any mammals. It's a real probe, so it's a nice control for this system, and you can use this to threshold for background. So this is a paper that actually just came out this month in Neuron, where researchers from Garrett Stuber's lab were studying the habenula. They prepared a single-cell suspension from microdissected habenula, and they generated a single-cell RNA sequencing data set profiling, in the end, more than 11,800 cells. And they used HiPlex to connect the single-cell RNA-seq data back to the tissue morphological context and spatially map the gene expression profiles of these cells. So habenula serves to regulate a variety of behaviors related to motivation and decision-making, but little is known about genetically defined cell types in this complex. Here we see clusters corresponding with cell classes, neurons in red here versus glia, for example, that were pulled out from this data set. Their relatedness, the relatedness between the clusters, and this file-in plot shows expression levels of canonical marker genes in neuronal and non-neuronal clusters. This heat map here is displaying expression levels of all genes in each cell class. it was known that the habenula can be divided into medial and lateral portions. For example, neurons in the medial habenula play a critical role in regulating nicotine reinforcement and anxiety and fear-related behaviors. Here in this UMAP plot, we're seeing just the 12 neuronal clusters that correspond with either medial or lateral habenula neuronal clusters, and they are clearly delineated based on expression of markers, including TAC2 and protocoadherin 10. Here, this is RNAscope data for using probes for these gene targets, and you can clearly see this anatomical delineation of medial versus lateral habenula, and then this is a marker that's common to both sides. And they created this really cool spatial mapping of gene expression patterns in medial and lateral habenula. So here we're actually looking at HiPlex data, and these HiPlex data are showing spatially biased distributions of gene expression. They showed that some transcriptionally defined clusters have correspondence with anatomically defined subnuclei, while other clusters distributed across subnuclei. In the supplemental data, you can actually find these representative images of all 12 genes that they detected, that they used to generate their spatial mapping data set. All 12 genes here on the left were stained in one single slide, and all 12 on the right were stained on a second tissue slide. So what's great about the HiPlex assay that I'm sure you can appreciate here is that it preserves a lot of tissue. So whether you're working with really precious samples or you're working in a very small structure where even a couple of microns makes a big difference, you know, a couple sections later on the cryostat, you're in a completely different region. HiPlex allowed them to visualize all of these targets while preserving the number of samples that were tested. And they go on to do some really cool things. They further characterize neurons in this complex. They use RNAscope probes to show transcriptionally defined populations that respond to aversive stimuli. It's an impressive resource. To do it justice, I'll just refer you to the paper itself. The methods are descriptive and easy to follow. A second paper that I wanted to talk about was this one published in 2016. So this was actually published before the HiPlex assay was developed by ACD. Here, Ozgoka et al., working with Stephen Quake's group, they identified novel subtypes of D1 and D2 medium spiny neurons in mouse striatum. But in this paper, they didn't actually show spatial relationships between D1 and D2 medium spiny neurons. They just have their single cell sequencing information. And so looking at this paper, what makes this a really great data set for validating with HiPlex is that the gene signature that defines the D1 medium spiny neuron subtype is comprised of 8 genes. So you can easily get at it with the HiPlex 8 reagent kit. And likewise, the D2 medium spiny neurons are also pulled out by a panel of 8 genes. But then 2 of these genes are common to both subtypes. So with a panel of 12, with the HiPlex 12 kit, you can spatially resolve all of these subtypes in just one single slide. So ACD pulled this paper, generated probes against the genes of interest that came out from the single cell data, and then ran a panel individually on a sagittal mouse brain tissue section, one slide for this 8-plex, one side for this 8-plex, and one side for this 12-plex. And here's how the data look, where here we're looking at that panel of 8 genes that pull out the DRD1A subpopulation. As indicated, this isn't necessarily showing every single cell that's pulled down by this population, but it's a representative image. Likewise, the DRD2 subpopulation with its panel of 8 genes. And then the 12-plex assay for all 12 gene targets that pull out different subtypes in striatum. Now, what you're looking at here is 12 colors all piled up on top of each other in the same image. So it starts to get a little bit difficult to look at. You combine 12 different colors, they all sort of start to turn white in the end. With the HiPlex registration software, one, you're registering images from your various rounds of HiPlex probe detection onto one another, but this also allows you to visualize these targets in a flexible manner. You can toggle on and off different channels. So for instance, if you just want to look at 2 genes together, DRD1A and DRD2, for example, you can do that. And here, what's kind of cool is, canonically, you think of DRD1A and DRD2 being mutually exclusive, but it was previously reported in the literature that there are cells that express both of these markers. And just looking at this HiPlex data in this panel here, you can see these 3 cells that are expressing both green dots and red dots, corresponding with co-expression of both of these markers. So those are 2 examples of neuroscience applications using the HiPlex. HiPlex is being used in other areas of research as well, not just mouse brain. Off the top of my head, researchers at Duke, for instance, are using HiPlex to study the tumor microenvironment, developing panels of genes that might be used to stratify patients for treatment or even used prognostically. So it's an interesting bunch of research that is coming out with the HiPlex. Regardless of your area of research, the first experiment that you should always run is to take 2 slides of your sample, run positive control probe on one and negative control probe on the other. And this serves several functions. You should get strong staining with positive control probe. The positive control probe is a cocktail of several different housekeeping genes at a variety of levels of expression. You'll have relatively low, medium, and relatively high expressors. But as housekeeping genes, they're constitutively expressed. So you should see signal in almost every cell in basically every tissue. And the negative control slide, as I mentioned, is a real RNAscope probe, but it's against a gene that's just not expressed in tissue in mammals. So once this looks good, once you see strong positive control probe staining and a clean negative, you can go ahead and run your target probe. But if this doesn't work out, if you're not seeing strong positive control probe staining, one thing you might need to do is optimize your pre-treat conditions. Worst case scenario, if the tissue wasn't prepared in such a way that the RNA quality is preserved, you may need to go back and collect fresh samples. On the note of pre-treat conditions and optimal digestion, I think this is easier to explain looking at just single-plex brown RNAscope chromogenic assay. Here, shown in left, is control probe PPIB, positive control probe PPIB, and on the right is negative control probe DAPV. The nuclei here are counterstained with hematoxylin, so they're purple. And again, this is single-plex brown RNAscope, so every dot corresponds with a transcript of PPIB. This is the kind of digestion we like to see in a tissue sample where the tissue morphology is a little bit softened almost, but you're getting a good amount of positive control probe staining and you don't see any signal here. However, if you go too far and you over-digest the tissue starts to look kind of smushy and falling apart. You actually start to lose RNAscope signal as well. Because the tissue architecture is kind of falling apart at this point, the signal is sort of clustering up and you have less signal than you had in optimally digested tissue. The nuclei are now undefined. However, you may not digest quite far enough. Here, the tissue morphology is still entirely intact. So while a pathologist might look at this and say, that looks fantastic, I can see so much about this tissue architecture, there's hardly any PPIB staining. You can see in this portion of the sample, there's a couple of really faint, hard to see small brown dots that are starting to appear. But this sample just wasn't digested more harshly enough that probe was able to access the RNA in the sample. Something else to keep in mind for fluorescent RNAscopes like HiPlex, over-digestion can cause autofluorescence to be even worse. So this is fresh frozen mouse brain tissue that doesn't have a high level of autofluorescence to start with, but in over-digested tissue, the background starts to come up, interfering with your readout. So this is optimally digested tissue. You can see the background's nice and clean, and you can clearly see 2 colors corresponding with 2 gene targets. The control probes are pre-mixed, ready to use at 1x concentrate. And these control probes, like the other HiPlex reagents, come in these little dropper bottles. So it's really easy to drop these reagents onto your slide. You barely even need a pipette. The 12-plex positive control probe, here I'm showing the human-specific control probe. You'll want species-specific positive control probes. There's a mouse version of this as well. 12 different housekeeping genes each labeled with a tail that will send it into a different fluorescent channel at the microscope. The negative control probe is 12 different probes, so 12 different tails, but it's that same gene. The other reason to use your positive and negative control probes, in addition to checking for RNA quality of the sample and for checking that your pre-treat conditions are optimal for your tissue type, is in cases where there are certain types of autofluorescence that you want to watch out for. Here we're seeing fresh frozen human brain tissue. This is the human-specific HiPlex 12-plex positive control probe. This image is from round 1 of the HiPlex assay. Because this is fresh frozen tissue, we wouldn't really expect to see a lot of autofluorescence from fixation, for instance. And by the way, this image is courtesy of a lab associated here in the Boston area with the Harvard Medical School. This is the negative control probe on a different tissue slide from this same sample. And what you can appreciate here is it looks like there is RNAscope signal, but we know that this is not real RNAscope signal because it's the negative control probe. This signal that we're seeing here is intrinsic to the tissue itself, and it's coming from an autofluorescent molecule called lipofuscin. On the chart on the right here, this brownish line, this sort of broad emission profile corresponds with the emission spectrum of lipofuscin. It just accumulates over time in lysosomes of postmitotic cells, such as neurons or cardiomyocytes, for example. The exact mechanisms of its accumulation are unclear, although it often correlates with aging. And there's just not much we can do about this intrinsic autofluorescence. So it's really helpful to have your negative control for imaging to make sure that you know what's true signal and what's not. So to summarize sort of our best practices for imaging fluorescent RNAscope, use your positive and negative controls to threshold for what true RNAscope signal is. It really comes in handy, especially at the fluorescent microscope. What you want is to find the highest dynamic range possible without overexposing your sample. And the best way to do that is focus in the DAPI channel and then find the optimal exposure time for the first channel of interest on your positive control probe slide. Exposure times really depend on your imaging system, so they will change depending on what your system actually is. But I just put up these examples to illustrate that you shouldn't need a very long exposure time. If you're needing to expose out to 800 milliseconds or a second long, something's wrong and you don't have real signal there. And then whatever exposure time you determine is optimal for that particular fluorescent channel, now check your negative control slide and make sure that the sample is not overexposed, that there's no signal detected in that same channel. If so, lower the exposure time. Although, as we just saw, there are some types of autofluorescence that you can't just lower the excitation power away and drop out. So make a note of that. And then go back and check your other fluorescent channels, making any necessary changes. Sometimes researchers don't understand that after setting these initial imaging parameters, using the same settings for positive and negative control slides, target probes may require you to adjust imaging parameters to capture optimal signal, depending on the expression level of the gene. So here, for instance, target probe SLC32A1 is a higher expressor than MEIS1. These images were captured with different imaging parameters. You would need more excitation power to see this lower expressor. So if a researcher set their excitation power or exposure time just based on this SLC signal and then put this slide in the microscope, you might be missing this signal generated from this probe. However, if you image the MEIS1 slide with this SLC slide with the same excitation power you use to image this slide, this now would probably be very highly overexposed. So there is some optimization that you can do depending on the probe level of signal that you're seeing based on the gene expression, the real biology of the target. One more thing to just draw your attention to is that different fluorescent channels behave differently. As I already mentioned, the green fluorescent channel has the most autofluorescence, so we recommend to put your highest expressing probe target into that fluorescent channel to optimize your signal to noise ratio. As far as the other HiPlex fluorophores go, red, far red, and this Alexa 750, the farther out you get, the cleaner the channel is, although of course you can't see these channels through the oculars of the scope. You have to scan in the microscope before you'll see them. But the signal to noise ratio out here is really excellent. So consider these properties of fluorescent microscopes as you're deciding which genes you want to be labeled with which tails, your T1, T2, T3, etc. So I'm going to conclude this discussion of the HiPlex assay by showing you how to use the image registration software to generate the beautiful images that I've been showing you. And basically what the software does is it uses the DAPI nuclei from the different rounds of imaging, and it overlays images onto one another based on the DAPI nuclei. Here's just a crude schematic of what that software does. So for instance, if we decide that the cyan image here is our reference image, the HiPlex registration software is going to transform these other two panels such that now the nuclei are overlaid on top of each other. The software can align images that are only 70% overlapping, although you really want the most overlap possible because non-overlapping edges outside of this Venn diagram will be cropped away. The user manual that comes with the image registration software is really pretty detailed. It has screenshots so you can follow along, and there's really no substitute for just getting into the software and clicking around. But I'll take you through a demo of what the workflow looks like. If you go to the File menu in the registration software, click on User's Guide. This will pop up the PDF of the user manual. To highlight just a couple of tips, as you're acquiring your images at the microscope, name your files carefully because you're performing 2 or 3 rounds of detection, right? So you want to include something like underscore round R1 for round 1, underscore R2, such that you know which rounds of imaging each image came from originally. The other really important thing here is the software requires that images that you're trying to overlay are the same size, same resolution, same pixel by pixel dimension. So if necessary, use Fiji and crop your images to the same size. You'll also need single channel images. So from that composite image you capture at the microscope, separate the channels and save each channel as its own separate file. If you do a 12-plex, for instance, you'll have a total of 15 files, 12 for your probes of interest, and then 3 for the DAPI from each round of staining. And it's these DAPI images you use first to write your transformation matrix. So going up to the File menu, click on Open, find the single channel images that correspond with your DAPI staining from each of the rounds of imaging that you did, select them all, and add them to your dashboard. Once they're added here in the dashboard, you're hitting the checkbox next to each of these files, go up to the Register menu, and select Register DAPI Images. What this does is whichever image is listed first in the dashboard, this is your reference image, and it will move the images lower down onto your reference image. So when you select that Register DAPI Images option, you'll see a dialog box pop up, and it will tell you which is your reference image and which image it's moving to align to your reference image. It will ask you to save the transformation matrix that it has just written. The program will suggest a name. Just save it with some name that you know which round of staining it came from. And once the registered image is created, you'll see it appear in the dashboard highlighted in green with an underscore R after the name for registered. Now, what you'll do is create a transformation matrix that moves your round 3's DAPI onto round 1's DAPI. So again, you see in the dialog box, it will tell you which is the reference image here, which one is being moved. And again, it will ask you to save the transformation matrix file that the software will write. And once the image is registered, again, it appears here in green with underscore R. And you can check the alignment by toggling on and off the R1 DAPI image that served as your reference and the registered images from R2 and R3, and they should overlap perfectly. Once that's done, you can remove the DAPI images you don't need, because keeping all of these images of DAPI creates redundant DAPI signal that you don't need. You don't need them, they've served their purpose at this point. But now it's time to align the other channels from round 2 and round 3. So if you go up to the File menu again, select Add Images to Current List. Selecting Open here will clear the files from the dashboard. You want to add more images, not start a new one. And you'll find and select just the probe channels from round 2. At this point, you can surely appreciate why it's really helpful to name your files such that you know from which round of staining each image came from. Once the round 2 probe channels are added to the dashboard, go up to the Register menu, and at this point, you're choosing Apply Registration Transform. You'll select the .tsm, the transformation matrix file that was written, to overlay the images from round 2 onto round 1. Hit OK. And voila, the R2 channels have now turned green and been replaced with _R for registered. And then you just repeat that process to overlay the images from round 3 onto round 1. So again, going up to the File menu, Add Images to Current List, select your channels from round 3. So now this is probe T9, T10, T11, and T12. Add them to your dashboard, apply Registration Transform, select the transformation matrix that was written originally by registering round 3 DAPI to round 1 DAPI, and hit OK. At which point, these are replaced with green, highlighted, and R_ files, indicating that they've been registered now and aligned to round 1. Now, whatever happened to the staining from round 1? Well, remember, we used the round 1 DAPI as our reference image in the first place. So you don't need to write a transformation matrix to align the probes from round 1 to their own DAPI. They're already aligned, but you do want to add them to your dashboard. So go up to File, Add Images to Current List, select the probe channels from the round 1 of staining, and now they're added. And you can crop away the overhanging edges by selecting all of the files in the dashboard, going up to Register and selecting Crop Overlapping Region, at which point the file names are replaced with _C at the end, and they're highlighted in yellow to indicate that these now have all been cropped. And at this point, again, remove the redundant DAPI, which you don't need if you haven't already done so. And here, the software allows you to pseudo-color each channel whichever color you want for easy visualization of these different markers, depending on who you want to look at together at any one time. The software, while really great at overlaying these images onto one another, and you have all of these color choices, it's really not designed for downstream analysis. You want to use this software to overlay the images, but then the better option is to use Fiji or a different downstream software to generate publication quality images. So at this point, you have a couple of different options for how you want to save your images, but I recommend to save them in grayscale for manipulation with different softwares downstream. Of course, you never want to adjust brightness and contrast too much. Hopefully you took a fairly honest image on the microscope in the first place, but using Fiji for that is the better option rather than using the HiPlex image registration software. Just one more note, I think I mentioned at the very beginning, we don't recommend HiPlex for really highly autofluorescent tissues like formalin-6 paraffin-embedded samples. The reason is the level of autofluorescence in these samples is so great that while the HiPlex assay has a great signal-to-noise ratio, it's not optimal for overcoming so much autofluorescence as you see in FFPE samples. However, we have a chemistry, a fluorescent RNAscope multiplexable chemistry called the Z2 chemistry that actually uses pyramide signal amplification fluorophores. And with this additional level of signal amplification, you actually combat the autofluorescence in FFPE samples, and you can use this chemistry to look for gene expression in FFPE samples without too much of a problem. Here's pictures of how the HiPlex reagents are kitted together. You have your pretreatment reagents. There's a couple of different flavors of protease. With this kit comes protease 4 and protease 3. In terms of relative strength, protease 4 is harsher, stronger than protease 3. There's also a target retrieval buffer. If you have fixed frozen tissue, you'll need to do a short, just 5-minute boiling step with target retrieval buffer to retrieve the RNA from the crosslinks formed by the formaldehyde fixation. A HiPlex detection kit by default comes with the 8-plex capability, and this comes with cleaving solution that you use to cleave off the fluorophores in between rounds of imaging. There's a wash buffer you'll use in between all of the hybridization steps to wash away any unbound oligonucleotides. If you're looking for 12 targets, you'll then also want the ancillary kit that allows you to detect 12 targets. And it comes with more cleaving solution because you'll do another round of cleaving than you would with an 8-plex. And then the probes. The 12-plex control probes come pre-cocktailed, pre-mixed at 1x concentrate, ready to go. Your target probes will all come at 50x concentrate in little Eppendorf tubes just like this, and you'll use the HiPlex probe diluent. This is basically a buffer to dilute your concentrated target probes 1 to 50 for application to the slide. Again, most of the kit reagents come in these little dropper bottles. It makes the kit so easy to use. The one thing you'll really need the pipette for is to do this dilution factor. Other equipment that you need, a hybridization oven where all of the hybridization steps occur. You need an incubator that holds temperature really reliably and holds humidity pretty precisely. So watch out. If you have a really old, large, decrepit incubator in lab, highly recommend try out the oven that ACD actually designed to work with this assay. The internal chamber is really small. It holds temperature reliably and stably well. The other thing that comes with that is this locking slide holder. All of the slides sort of lock into place. This fits 20 slides at a time, and it allows you to move all of your slides together at once from the hybridization tray, where the hive steps occur, into wash buffer. Here is this clear plastic wash tray where the washing steps happen. And with this locking slide holder, you just move all of your slides together at once, back and forth. It makes the workflow really easy, and it reduces any slide-to-slide variability you might have from taking slides out one by one and putting into a Kotlin jar, for instance. And then the only other thing you would need if you have fixed frozen tissue is a steamer. We recommend using a steamer for the target retrieval step. It tends to be gentler on tissue, but you can also use a beaker and a hot plate if you don't have a steamer, or, you know, it's really pretty cheap on Amazon. There's a particular steamer that we recommend to people who don't have a steamer in lab already. And that's about it. The only other thing I wanted to mention is that you can, in fact, do RNAscope and immunostaining together on the same slide if you're interested in looking at both transcripts as well as protein. What we recommend, though, is to do your antibody staining at the end, because RNAscope is, or RNA is an unstable molecule. RNAscope is a really powerful assay for detecting even degraded RNA, but do yourself a favor, detect your RNA first by hybridizing your probe, amplifying your signal, and capturing that signal, and then bind your antibody at the end. So to summarize, RNAscope technology uses both a unique probe design strategy as well as the signal amplification to dramatically improve the signal-to-noise ratio over what you get with traditional in situ. And it's pretty easy to design a new probe if we don't have one in the catalog that meets your specifications. It's really sensitive thanks to that amplification system. You can multiplex. With the HiPlex assay, you're detecting up to 12 targets at the same time in one side of tissue while maintaining single-cell resolution. We have an assay called BaseScope. If you're interested in alternative splicing or really short sequences or even point mutations, we have a probe and a platform for that. It's called BaseScope. I am happy to talk about it more. We have actually webinars available, pre-recorded, that you can learn more about how to use BaseScope to detect these sorts of RNAs. And just a pretty exciting announcement, we have an assay coming out next month where you can detect sequences as short as like 22 nucleotides. If you're interested in mature microRNA, or if you're using ASOs or silencing RNAs, really, really short RNA species, are soon going to be detectable with microRNA scopes. And you can combine RNAscope, Spatial, whatever RNAscope type of chemistry you're running with an immunostain at the end if you're interested in looking for RNA and proteins together. With that, I hope I haven't bored you to death. Thank you so much for your attention, and I'm happy to take any questions that you might have.
Thank you, Emily. What an excellent presentation that was. We did have quite a lot of questions come through, and Emily's been working her way through those. I really hope you enjoyed today's presentation. I do have a couple of questions for you live, Emily. Let me go ahead and pull them out for you. The first one here, let's see. After cleaving, can you go back and detect the same probe again in a later round? Emily, let's see if I need to unmute you. Oh, you are unmuted. You seem to be unmuted. Can you hear okay? Okay, let's see. We seem to be having a little difficulty getting Emily back on mute. If you're able to answer in the chat, if you can let me know, and then we can go ahead and maybe conduct the Q&A from there. Okay, perfect. Great. Let's see. So the first question was again, after cleaving, can you go back and detect the same probe again or in a later round? Let's see. Emily is going to be typing her answers in the chat for us here. Her response is you cannot redetect same probe again in one HiPlex experiment. Thank you, Emily. Second question, more information. Once the fluorophore is cleaved away, the site of a fluorophore would attach to it, so then it becomes blocked. Thank you, Emily. Second question, can you do an image?
Can you hear me now?
Oh, we can. Yes, perfect. Excellent.
All right. Sorry about that.
No worries at all. Thank you for that. There are quite a few questions that you've been addressing in chat already. I have a second one here for you. Can you do an immunostain with HiPlex?
You can. So we would just recommend that you do that at the end because RNA is an unstable molecule, especially as compared to protein. Detect your RNA signal first, but then at the end of the assay, you can decover slip one more time. You can cleave off the fluorophores one last time, and then you've essentially got a blank slate. And you can go ahead and use that tissue and attach a protein. Now, the one caveat is sometimes the pre-treat portion of the RNAscope assay that we need to get probes access to their target RNA can mess with epitopes. An antibody would then be needing to recognize. But we have like a number of publications out there now with antibodies that work just fine. And there's optimization we can run, additional reagents we can supply to help the success of that dual stain-ish IHC.
Great, thank you. Another question here for us. If I have formalin fixed paraffin embedded tissue, can I still use HiPlex?
So we don't recommend HiPlex for FFPE tissue. The RNAscope assay, the HiPlex amplification system is good. It's really sensitive, but because FFPE tissue is so highly autofluorescent to start with, your signal to noise ratio just isn't that great, especially in that green channel. So if you have FFPE, we recommend the version 2 multiplex RNAscope chemistry instead. With that one, you look at up to 4 targets on the same slide. So that's where we would point you to instead of using HiPlex for FFPE.
Okay, Sounds good. Somebody actually put into the chat, you didn't get a chance to answer that one. Do you have any suggestions on capturing the same spots for imaging? They're using EVOS, but there's a certain amount of wiggle room, so often they don't overlap. What's your proposal?
Yeah, great question. It's great if you do have a microscope that has an automated stage, because then you can save the coordinates of the image that you take, and as long as you load the slide into the same orientation on the stage each time, the stage will just zoom to where you were before. But I would say that's less common than having a stage that's not automated. And in that case, your best bet is to find an area of tissue architecture that you can come back to each time. Some types of, so the dentate gyrus, for instance, in central nervous system tissue is a really good landmark. You're trying to find a landmark in that DAPI channel that you can come back to again. I like to draw a little Sharpie circle on the bottom of the slide, roughly around the ROI. Obviously, you can't see that through the oculars of the scope or when you're scanning, but it can help you kind of roughly get to the same spot. And the last thing to consider, the HiPlex registration software needs images that are only about 70% overlapping. So it's okay to have a little wiggle room. It's okay to be just slightly off from the ROI that you took an image from originally. You can still overlay those images. So yeah, it's definitely a consideration to, have to go back and find the ROI again. Of course, if you have a whole slide scanner, that circumnavigates the entire issue, because you just take a picture of the entire slide.
Yeah, great answer. Thank you so much, Emily. And thank you everybody for the questions that you came in. We did have, let me see, one more actually. Kate came back. They have an automated stage and they've used the ACD software, but it doesn't work well. So perhaps, Kate, let's reach out to support, and if you can send a message to me, we will get you in contact with Emily and we can do some proper troubleshooting for you.
Just gonna say, I mean, that's why I exist, I suppose. You have a tech support team that can help you with that. And so in ACD's in-house lab, we also use a whole slide scanner and register images through the HiPlex registration software that way. So yeah, let's talk, because I'm sure we can figure it out, like what's not working well and work around that.
Fantastic. So we're coming to the end of the presentation. We're almost out of time. And I was just wondering, shameless plug here, Emily, can you talk about an upcoming product that we've got coming called the HiPlex Up? It's a new exciting thing. Tell us a little bit, give us a sneak preview of what's happening with that.
Essentially, it's a way of repeating this 12-plex assay I talked about today. Again and again. We've tested up to a total of 48 targets. So once you do this HiPlex assay once through, you do a step where you then wipe off the probes and the trees. That doesn't happen in a typical 12-plex HiPlex. You're just removing the fluorophore. But with HiPlex Up, you would then do a step where you remove everything, sweep that slate clean entirely, and then reprobe with your next set of probes. And it's funny because the paper I talked about, the Hashikawa et al. Paper from Garrett Stuber's lab, they did something similar before we actually developed this new assay. So that was a really cool proof of concept that it does work pretty well. But now we do have the official ACD reagents that are optimized to work. So HiPlex Up is the name of the product, and I guess you would just reach out to your account manager for more information on how to get a hold of those reagents.
Sounds fantastic. Yes, and it's coming soon. So please watch this space. Please reach out to the account managers, check our website. We'll have some new information coming out very, very soon. So once again, thank you, Emily. What an excellent presentation. Great questions from the team here. We've had a really good, engaged audience today, so we really appreciate it. On behalf of the West Coast team, Kristen, Jimmy, Abby and Dijon, We hope you enjoyed today's presentation, and they will be doing a few more coming up over the next few weeks. So please make sure that you check out our acdbio.com events page and register for the next upcoming event. And with that, I'm going to end today's presentation. Thank you very much.